Assalamualaikum. This is the second part for enzyme immunoassay. So this part we will have a look on the principle behind the procedure of enzyme immunoassay. So this is the first step. Primary antibody or antigen binds or coated to solid surface. For example, plastic. So they will, when a process where they will be coated to the solid surface. Here we can detect any antigen that we want or detect any antibody being produced in the sample. So if we want to detect an antigen, of course we need to first stick the antibody, specific antibody against the antigen that we want to look for. For example, if we want to look for the virus COVID-19, we need to stick the antibody against COVID-19, not any antibody, but antibodies specifically against COVID-19 to the solid surface. And if you want to look for example, patient that already been infected with COVID-19, you want to know whether they have an immune response, they produce antibodies against the infection. So we need to put the antigen that we want to look for here. For example, if we want to look for antibody production in patient against COVID-19, we place here the COVID-19 virus particle or antigen. This is the first step. And then the second step, we add the sample. So again, we are looking for the antigen. We have the specific antibody for what we are looking for. So now the patient should have the specific antigen against the specific antibodies. And if you want to look for antibodies being produced by the patient, the sample should have the antibodies against the antigen. They need to be very specific against each other. And of course, if they match, they will bind to the primary antibody. We call this as the primary antibody because they are the first to be put in the reaction, or the primary antigen. But remember, there are still antigens or antibodies. They are not able to bind because the limited capacity of the antibodies or antigen on the solid surface. So what to do? We need to wash away the remaining unbound antigen or antibodies. One of the buffer that can be used is phosphate buffer solution to wash away the unbound sample. We don't need the unbound sample to be round because we don't want them to react with the conjugated antibodies. On the fourth step, we will add the antibody enzyme conjugate. These are antibodies with enzymes against the specific antigen or antibodies against human antibodies. These are the antibodies, specific antibodies earlier that bound to the specific antigen. Imagine that if the unbound sample is still around, these antibodies, instead of binding to the appropriate place, they will bound to the freely available antigens or antibodies. And of course, the antibody enzyme conjugate will bind to their specific antibody or antigen. In this case, conjugated antibodies against the specific antigen that we are looking for, or antibodies against human antibodies, because earlier we put the specific antigen. We want to detect the patient antibodies, the specific antibodies against the specific antigen. So we need to use antibodies against human antibodies. One more time, the unbound antibody enzyme conjugate need to be washed away by using PBS. They need to be washed away because we don't want them to react with the substrate that we're going to add later in the 6th step. So on the 6th step, substrate will be added, and the enzyme, respective enzyme for the specific substrate is available. They stick to the reaction earlier. And if the enzyme present, enzyme in the conjugated antibodies will convert substrate to colored product. So we can observe the production of color. This means that this patient is positive of having the specific antigen in their blood. For example, the COVID-19 virus particles in the patient blood, or the patient have antibodies produced. I mean, probably they are immune to the antigen. For example, COVID-19, they have the antibody against COVID-19 here. So these conjugated enzyme antibodies act as an indicator. So how the result and interpretation? Positive sample will have color changes, and no color changes means negative samples. If not present, we're talking about the enzyme antibody conjugate not present because they have been washed away earlier. Substrate will stay colorless because there is no enzyme to turn them to become active and produce color. What happened here? This is a negative samples. This patient do not have the antigen in their blood. So that means earlier we put the specific antibodies, let's say against COVID-19, but the patient doesn't have an infection with COVID-19, so there is no antigen of COVID-19. The virus is not available in their blood, so it will not stick to the primary antibody. Even though we put the conjugated antibodies, conjugated antibodies doesn't have any place here, doesn't have these antibodies to stick, so they will be washed away, and in the end when we put the substrate cannot react because there is no enzyme available. The same thing goes when detecting antibody. We place the antigen, specific antigen that we are looking for. This antigen can be commercially produced. we coat them to the solid surface. We want to look whether the patient blood contain the antibodies against this infection, this antigen, but somehow the patient do not have the antibodies, do not produce the antibodies. So when we put the conjugated antibodies with enzyme, they cannot stick to the human antibodies because they are not around. In the end, when we added the substrate, there is no place for substrate to react because there is no enzyme available. So we can observe by no colors changes. For results and interpretation, for EIA, for example, ELISA, we commonly use a microplate reader, a type of photometer that can determine the level of absorbance. The advantages of EIA includes they can detect against a wide variety of antigens. That means they are so versatile. They can be used to detect a lot, huge numbers of organisms, virus, bacteria, parasites, fungi, hormones, exposure to disease. That means we can detect the antibody against the disease. Food allergens, for example, antibodies against eggs, against peanuts, and etc. They are easy to perform and they are quick. The disadvantages of EIA includes because we are dealing with enzyme activity may be affected by the plasma constituents. The substance within the patient serum or plasma might react with the enzyme itself, so we can have wrongly positive or false negative results because of the unwanted reaction between the enzymes. And enzyme also known to decrease in their reactivity over time. So again, the limitation come from the properties of the enzyme itself, and they are costly or expensive because we need to buy the reagents. The reagents quite cheap actually, but the antibodies is very expensive. The equipments, so that's why they are considered expensive. Thank you and have a nice day.